Hello everyone. Today I'll be discussing the contents of week 5, following with some questions and some experimental techniques related to that. So, in the previous weeks, we have learned about how we have learned about classification of different organisms into different groups, how it pertains to evolution of those organisms, and how are these two things related. uh, Basic structure of a cell, what are the different constituents that make up the cell. In the past week, we learned about various biomolecules, some of which are DNA, RNA, lipid, and carbohydrates. In this week, we'll be learning about proteins and different aspects of proteins. Okay, so coming to proteins, these are essentially polymers of amino acids, and the bond between two amino acids is known as a peptide bond. Okay, and this is a covalent linkage. This is not a it's not any other type of bond which is temporary. This is a covalent bond, implies that it requires a very high energy to break this structure. So the basic, like nucleotides were the basic unit for DNA and RNA. For proteins, the basic unit is amino acid, which has a structure like this. So it has a amino group, it has a carboxylic acid group, it has a hydrogen atom and a side chain, and depending upon what the side chain is, the amino acids are going to be different. Because these all of these groups are different, all of these groups attached to the so this carbon is known as the central carbon. Because the groups attached to the central carbon are different, this carbon, or the alpha carbon, is usually chiral in nature, except for in glycine. So in case of glycine, the side chain is, the R group is on hydrogen atom, because of which there is no chirality in that, but we'll come to that later. So each of the protein can be broken down into its constituent amino acids by a variety of methods to study the free amino acids, and 20 different amino acids are found in any protein. Like, a protein can be a combination of these 20 basic amino acids. So all of the 20 amino acids are alpha amino acids, which are with a common structure, which is this amino group, carboxylic acid group, side chain, and the hydrogen atom. So, and the difference comes from the side chain here. The side chains might be different for different will be different for different amino acids. And because of the chirality of this carbon, you can chirality of this carbon, there are enantiomers. uh, we can find stereoisomers of each amino acids, each chiral amino acids, which is known as enantiomers. And these are essentially superimposable mirror images of non-superimposable mirror images of each other. If you were to place a mirror here, you get a mirror image of this in the enantiomeric counterpart. Okay, please stop me if there are any questions. So the there are 20 amino acids, and the amino acids and the nomenclature of amino acids is done by two methods. Either it is denoted by first letter, or by a single letter. This single letter uh, single letter nomenclature might be or might not be the first letter. So in cases like glycine, alanine, histidine, all of these cases, the single letter nomenclature is the first letter of that amino acid. Whereas cases like arginine, lysine, glutamine, in those cases it's a different letter, because the other letters were already associated uh, with the with other amino acids. The first amino acid to be discovered was asparagine in 1806, and earlier the nomenclature of amino acids was either based on the structure, such as glutamic acid, aspartic acid, or based on where it was isolated from first. For example, asparagine was isolated from asparagus, hence the name asparagine. Glutamate was from wheat gluten, tyrosine was from cheese. The Greek name of cheese is tyros, and glycine was named so because of its sweet taste. So as you can see, there is the common thing amongst all of these is these three moieties which are common among all of these, and this particular group is the thing that is different, and depending on what this is, the amino acid is different. The other nomenclature is a three-letter nomenclature, wherein it's usually the first three letters of the amino acid. So for arginine, it's ARG, for glycine, GLY, but for certain cases like glutamine, it's GLN, because glutamic acid was named GLE. Okay. Um, one more thing is, so this is the ionic form of the amino acid. So earlier, whatever structure you saw was this is the neutral structure. If the side chain is also neutral, this can be called as the neutral structure. However, if the amino acid is placed in a polar solvent, then you get something like this, wherein there is a, because of the basic nature of the amino group, it accepts a hydrogen ion, and COH group, because of its acidic nature, releases a hydrogen ion. This is only if they are put in a solvent, a proper solvent. If they are not, then they will have a structure like this. Okay. So depending, because the structures of these amino acids are different, the molecular weights are going to vary, but on an average it is assumed that if you don't know the structure of the amino acid sequence, if you don't know the sequence of the amino acids, it can be assumed that each amino acid has a weight of 110 daltons on an average. If you know the size of the protein, then you can compute the number of amino acids contained in that protein approximately, depending on depending on this value of 110 daltons. So for example, if there is a protein which is 22 keda, then that implies it's 22,000 daltons. That if you were to take a 110 size of each amino acid, then you will get 200, you will get a string of amino acids approximately of 200 length. Okay. So depending on the R groups, all of these amino acids can be placed in different kinds of groups. Non-polar, aliphatic, aromatic, polar, negatively charged, and positively charged. Non-polar aliphatic amino acids are amino acids which are non-polar and hydrophobic. Examples are alanine, valine, leucine, isoleucine, glycine, methionine, proline. Because of, suppose you take alanine, because it's a CH3, it's a non-polar group, therefore it is called a non-polar amino acid. Similarly, similar thing goes for glycine, where, because it's an H group, it's a non-polar group. Okay. There are certain aromatic amino acids in which the R group is a hydrophobic side chain. Specifically, usually these are R groups which have a benzene ring in them. Examples are phenylalanine, tyrosine, and tryptophan. So if you go here, you see that phenylalanine has a benzene ring and a CH2 group, tyrosine and tryptophan. All of these have a hydrophobic side chain because of attached to a methyl moiety, because of which they are named as aromatic groups. Polar uncharged R groups. These R groups are uncharged, but they are more polar than the hydrophobic amino acids. So the R group are, if you were to put it in a polar solvent, they would dissolve better. Examples are serine, threonine, cysteine, asparagines, and glutamine. Some R groups have a net negative charge because they are acidic in nature. Examples are aspartate or glutamate, essentially aspartic acid and glutamic acid. So these two, because of the COH group in the in place of the R group, these are acidic in nature. On the other hand, lysine, because of the NH2 group, is a positively charged amino acid. So R group are, in case of positively charged amino acids, the R group is basic with a net positive charge. Examples are arginine and lysine. So how do you know what amino acids are present in which of the in a particular sequence of amino acids? So one of the techniques that is used to analyze the amino acid constituent is thin layer chromatography, which is essentially a chromatographic method wherein, so any chromatographic method has two basic components. One is a stationary phase and one is a mobile phase. In this case, the stationary phase is silica or alumina, which is plated on a glass or aluminum foil as a thin layer, and the mobile phase is a liquid which is allowed to run through the stationary phase. In this case, in case of thin layer chromatography, what happens is a foil with the stationary phase coated on it is placed in a beaker which has some solvent, and depending on the solvent, the distances that the amino acids will travel will vary a lot. So the proper choice of solvent is very important in case of thin layer chromatography. So in this case, because there is a solvent at the bottom, by sheer diffusion, the mobile phase or the solvent runs from bottom to top. However, in most other chromatographic techniques, the mobile phase runs from top to bottom by gravity or a pump. So usually what would happen is you have a column of sorts in other chromatographic methods, and you have your stationary phase here. You put your sample in here, and because of gravity, the sample flows downwards, and they can then collect your elute in another tube. But in case of thin layer chromatography, the stationary phase absorbs the solvent, and depending on the interactions of the amino acids with the stationary phase and with the mobile phase, they're going to run for a different length. So depending on the interactions, the stationary phase will retard the movement of the molecule, where the mobile phase will promote it, and the net movement is where you will place your where it will be placing your amino acid. So if suppose the force of the mobile phase is Fm and the retardation force by the stationary phase is Fs, then the effective force will be Fm minus Fs. In cases in case where Fm is equal to Fs, in the assumption where Fm is equal to Fs, the position will be controlled by the nature or the functional group present on the molecule or the analyte, the nature of the composition of the mobile phase, the thickness of the stationary phase. If you take a thicker stationary phase, then it will require more time to run through it, the functional group present in the stationary phase. So the composition of the stationary phase, the composition of mobile phase, and the composition of analyte, all of these three affect the forces that amino acid is going to feel. Is there a question? Okay. So what would you compute from this is, if this is what you are looking at, then the distance traveled by this spot once you are done running your experiment, divided by the distance traveled by the solvent, the total distance of the solvent front. The ratio gives you a ratio called as RF. This RF will, for a specific stationary phase and mobile phase, a particular amino acid will have a constant RF. So what you can do is, you can compare it to the known RF values and then predict what the amino acid is. So there are several steps required to perform the thin layer chromatography. First, you need to make the thin layer chromatography plate. Then you do the spotting, or you put your sample on the thin layer chromatography plate, so that it is absorbed at that point. Keep in mind that at this point there is no solvent yet, so the spot is going, the amino acids or the analyte is going to stay at the point where it was put. Then it is allowed to dry. You mark where it was put, and then what you can also do is, you can take a bigger TLC plate and you spot some molecules for which you know the constituents. Suppose pure glycine or pure alanine, you can spot that. So this gives you a reference of sorts. Is there a question? Okay. If this gives you a reference of sorts for how far has your analyte traveled and how far has this compound traveled, you can compare between this and then map out what is exactly your analyte composition. Once the spot is dried, it is placed in the TLC chamber, so in such a way that the spot is not below the solvent level, so it's slightly above. Then the solvent front is allowed to move until the end of the plate. So your distance will be the spot to the end of the plate. Then the plate is taken out, air dried. If you are putting a colored compound, then it will form colored spots at particular points, and you don't need to do any additional staining. If in case it's not colored, then you need to spray it with a staining reagent. 4x or ninhydrin are used to stain amino acids. Otherwise, what can be, if you don't want to stain the TLC plate, what you can do is, you can place the TLC plate with an X-ray film on top for 48 to 72 hours, and then you process the X-ray film. So this is because this technique is known as autoradiography. Or you can some compounds have their own fluorescence due to some structural due to their structure. So in presence of particular wavelength of light, they will fluoresce. So in particular, a lot of these compounds are very sensitive to UV light. So the TLC plate can be placed in a UV chamber and then visualized under UV radiation for spots. Okay. So we now know that proteins are essentially polymers of amino acids joined by covalent bonds. The bond that forms between two amino acid, between the carboxylic group of first amino acid and this amine group of the second amino acid, is a condensation or dehydration reaction, and this particular bond has a double bond character, due to which the rotations that the bond can go is very limited. So depending on the structure of the uh, depending on the bond between the NCA alpha and CC alpha, uh, there are particular angles that uh, these bonds can undergo. These are known as dihedral angles, and they are designated by phi and psi. Like each of the NC alpha bond is called as phi, the CC alpha bond is called as psi, and they are able to rotate for particular angles from between minus 180 to 180 degrees with some restrictions. And G.N. Ramachandran, what he had done is he had made tripeptide sequences wherein he studied how much each of these bonds can rotate, and made something known as Ramachandran plot. So all of these dotted areas are the areas wherein uh, dihedral angles can place. So this is the psi and this is phi. So the prediction, this is sort of used like a prediction tool to validate the 3D structure of a protein. So the rotation angles can only form in these dotted areas. For some of the solid lines are the lines wherein it is known that there exist bonds, like well studied, that there exist bonds that rotate at these particular angles, but the dotted areas are also possible but not reported. Okay. So now that we know the basic structure of a protein, the basic building block of a protein, in cells the protein is not found in as a string of amino acids. Rather, it's found in a 3D conformation where it's folded into multiple structures. And so the string of amino acids, the sequence of amino acids, is known as primary structure, where it's just plain sequence of amino acids. Then these sequence of amino acids, depending upon the composition of the amino acid, can fold into various structures. These 3D structures, are known as secondary structures. These secondary structures can further fold to form tertiary structure. Now, this tertiary structure is a single subunit. Multiple such tertiary structures can arrange themselves to form quaternary structure, where different subunits can come together and make up a complex of protein. Okay, so 4 different levels of structures. The first level is primary structure, second is secondary structure, third is the tertiary structure, and fourth is a quaternary structure. So the amino acid sequence is essentially the primary structure, and the constituents of this sequence is predicts what the secondary structure is going to be. If if you were to compare two primary structures of the same length, they might not form the same secondary structure. That depends upon the individual constituents. So how do we determine the primary structure? This, like we did for if you remember, for nucleic acids, there are certain sequencing methods used to predict what the sequence of the amino acid of the nucleotides is, so that you have an idea of what exactly the sequence of your DNA or RNA molecule is. And there are certain methods to sequence proteins as well. So first, so if you were to go with a individual protein, not a mixture of proteins, one individual protein, you first need to break the bonds that stabilize the protein, so that you have a string of just a plain string of amino acids. You can then add different reagents to make sure that which amino acids are at what place. Okay. So the first stage of determining the structure is breaking the disulfide bonds, because this would interfere with the complete sequencing of the procedure, because it does not allow the release of cleaved amino acid from the peptide chain. So there are two approaches to disrupt the disulfide bonds. Remember, these disulfide bonds only form if there are cysteine residues within the protein sequence. If there are, because cysteine, if you see here in this chart, if you see here in this chart, cysteine is the only one which has sulfur groups in it. None of the others have it. So only in case if there are cysteine molecules, then free cysteine molecules, then you can you find sulfide linkages. How do you break this? You can do an oxidation reaction or a reduction reaction. If you were to do an oxidation reaction, so oxidation reaction means addition of oxygen atoms into you. Is there a question? Okay. Addition of oxygen atoms into your compound. Reduction means you either remove oxygen or add hydrogen atoms. So if you were to do an oxidation reaction, you do it by performic acid, which is there a question? Which will lead to a structure like this, which can then be analyzed further, or you do a reduction reaction, which you can do either by DTT or dithiothiol or beta mercaptoethanol or BME. So this will if you were to do it just by DTT, then you will have two SH groups, which can then be stabilized further by treatment with iodoacetate to form carboxymethyl lysine. This so if there is a structure like this may reassemble back to form a disulfide bond, but the carboxymethylated cysteine residues will deter the free formation of the disulfide bond. So once you have cleaved the disulfide linkages, then you can use various chemical agents targeting certain proteins or certain amino acids to see if there are specific recognition sequences. There is some disturbance. If there is a question, please ask. Otherwise, I would request you to shut the mic. Okay. If your mic is not working, you can post a question as well. Okay, fine. Okay. So, reagents such as trypsin, chymotrypsin, pepsin, cyanogen bromide attacks particular residues in the polypeptide sequence, and therefore you will have different overlapping short oligomeric sequences which you can analyze and then combine to know what is the sequence. So for example, trypsin only cleaves after lysine on arginine residues, chymotrypsin after phenylalanine, tryptophan or tyrosine residues, pepsin after leucine, phenylalanine, tryptophan and tyrosine, and cyanogen bromide after methionine residues. Then you overlap these sequences and then know what is the exact whole sequence of the polypeptide chain. Next comes sequencing of the peptides. So the first thing that you need to do is identify the N-terminal amino acid. So there is a N-terminal and there is a C-terminal, right? So you need to first identify this. This can be done by three processes, by a three-step process. First is you break down the first step is a chemical reaction, that is performed to label the terminal amino acid groups, such as you can do it by Sanger reagent, 1-fluoro-2,4-dinitrobenzene, DFNB, and dancyl chloride. These will label the free amino acid present on the base, free amino group present on basic amino acids such as lysine and arginine, to form a dinitrophenyl amino acid complex. This then you can do an acid hydrolysis. You essentially place the compound in a acidic solution of, and this leads to breakage of the dinitrophenyl and amino acid group from the peptide bond. So there is a dinitrophenyl amino acid complex in the solution and the rest of the peptide in your rest of the polypeptide sequence. Then you can do a HPLC or TLC separation of the complex to compare it. You can run a TLC, you can spot it with various reference solutions, and then you have your actual polypeptide sequence to know what the N-terminal amino acid is. And you can keep repeating this process to know what is the sequence. So if you have a 20 amino acid long polypeptide sequence, then you need to perform these reactions 20 times. Then the second method, that was Sanger sequencing method. The second method is Edmond degradation sequencing, wherein it's similar to Sanger sequencing. There is a reagent called phenyl isothiocyanate that reacts with the terminal amino group or the N terminal to form cyclic phenyl thiocarbamyl derivative, and this can be cleaved from the main chain to form a thiazolinone derivative. This can be extracted into an organic solvent to form a PTH amino acid derivative in presence of an acid, and this can, the PTH amino acid complex can then be identified by HPLC or TLC after comparing with a standard of amino acids. And this step can be repeated again to determine what the next amino acid is. So most of the sequencing methods focus on the N-terminal amino acid. There are not a lot of methods developed for the C-terminal amino acid analysis. The most common one is to treat the protein with carboxypeptidase, which releases the C-terminal amino acid, and then you test the solution. And this can be repeated multiple times. So what you are doing is sequencing the polypeptide sequence from the C terminal instead of the N terminal. So you can use different protein cleavage reagents, and you have basically obtained different stretches of amino acids which have some overlapping regions. You can then arrange these overlapping regions and compute what the exact sequence is. If to find presence or absence of disulfide bonds, you can cleave the protein with and without breaking the disulfide linkages. This will show you what is the difference between the two sequences that you got, and those will be the sites where disulfide bonds would have formed. Okay. Recently there are mass spectrometric methods have been used in conjugation with proteomics to find out what is the exact amino acid sequence. Let me just give a brief. Okay. So what you do is you extract the proteins. This can be a single protein that you want to perform mass spectrometry for, or this can be a mixture of proteins. So if you are expressing a single protein in a bacteria, overexpressing that protein, then purifying it, then you have a single protein that can be studied under mass spectrometry. If you have something known as a lysate, which is essentially a homogenized mixture of a cell, then you have a mixture of different proteins. So you digest these proteins into peptides. You mass spectrometry uh, functions with the mass and charge principle. So you charge these smaller peptides, then you put it uh, separate it depending on their mass and charges. You fragment the peptide ions, and then you take a mass spectrum, a spectral reading. Then you look at the relative abundance, the uh, overlapping sequences, and all of this will give you the exact structure of the amino acid of the protein. So mass spectrometry is used if you want to know what proteins are there present in a particular sample, how abundant these proteins are, what is the relative ratios of these proteins, and what is the sequence of these proteins. Okay, so this is mass spectrometry. This is very popular in research labs which work on determining what proteins are present in a sample, what are absent, to have a more wider range of proteins to study. So now we know what the primary structure is. What about the secondary structure? So there are particular structures that the amino acids arrange themselves in to form secondary structures. These are alpha helix, beta sheet, and turns. So alpha helix is a helical structure termed as alpha helix by Linus Pauling. This structure, in this structure, the polypeptide backbone is wound around a central axis with the R group of the amino acid protruding outward from the helix backbone. So it's kind of like a DNA structure. Most of the proteins have a right-handed alpha helix. Some might have left-handed alpha helices. Depends on the sequence of the amino acids. The other secondary structure that is predominantly found in proteins is a beta sheet, in which R groups protrude in a zigzag structure in opposite directions, giving an alternating structure. And the third thing that you would encounter are turns. These don't have any definite structure or any definite sequence of polypeptides associated with them. So both alpha helices and beta sheets, for both of these, there is a particular sequence of amino acids that will form these structures. But that's not true for beta turns. These are essentially connections between two beta sheets, two alpha helices, or one alpha helix and one alpha helix and one beta sheet. There is no consistency on the number of amino acids or their sequence. So using all of these three, a protein can obtain its 3D conformation. Next, the secondary structures will fold and condense to give a higher order of organization known as the tertiary structure. If multiple polypeptides are involved in the constitution of the protein, then the tertiary structure of these polypeptide chains come together to form quaternary structure. But this is only true if there are multiple polypeptides. Quaternary structure won't be seen if there is in a single protein subunit. Secondary and tertiary structures are usually determined by X-ray crystallography or NMR spectroscopic methods. Just let me. So what happens in X-ray crystallography is you isolate the protein, you crystallize it, then you irradiate it with X-ray. So depending on the orientation of the crystal, there will be different phases of the amino acids that you will see of the protein structure that you'll see. So what is usually done is the whole crystal is rotated for 360 degrees to find the complete structure of the protein. Then you uh, run an make a electron density map, which can then be compared to existing structures to find the final model or the structure. Next is NMR, or nuclear magnetic resonance. In this, so uh, in this you need a labeled protein, something uh, which is a has a C14 or deuterium in the protein itself, so that they can be used for NMR. The first step is to label the protein. Then you do the sample prep, put it in an NMR tube. Then you collect the NMR data, which will look something like this. Then you run the peaks, you run these, you do Fourier transforms, there is a lot of math involved to finally reach this stage which is signal assignments. Then what you do is you compare it with the existing library. De novo NMR is possible for new molecules, but it's slightly more difficult than comparing to existing libraries. If you have a NMR data, you compare that data with existing libraries to find what are the possible structures in that sequence. Then you uh, then there is a lot of post processing, validation, predict, and then you finally predict the structure of the compound. Or the other reason that NMR is used is to find out if there is an abundance of a particular analyte. For example, if you are looking at protein X, you run your NMR, you quantify how much protein X is there in the lysate, and you will have a standard of what protein X, you have a protein X standard, like a standard curve of sorts, and then you know how much protein X is present in different experimental conditions of yours. Does I hope that made sense. Fine. So these two methods can be used to determine the three-dimensional structure of the target experimentally. Other uh, methods uh, include homology modeling, wherein so there is a database full of known three-dimensional models of various polypeptide sequences. What you do is you take your polypeptide sequence, you match it to whatever existing polypeptide sequences are there to predict what is probably going to be their three-dimensional structure of the protein. So this is based on the idea that a particular amino acid sequence will fold in a particular manner always to form a three-dimensional conformation to get uh, to the basal state, which has the minimum free energy. Okay, but this is more of a comparative analysis than actually knowing what the structure is of a protein. To determine the quaternary structure, what you do is you run the protein. You can run a native page wherein there is no reducing agents used, or you can do a bunch of stuff and then figure out what is the size of the quaternary structure. Then you reduce the lysate. So reducing the lysate is essentially going to break down the component into its primary or secondary structure. Then you run the lysate in a acrylamide gel, figure out what the size of the primary structure is, and then you compare these two to find out how many of these primary structures combine to make the quaternary structure. Okay. Now what are the functions of the proteins? why do you need to understand what is the structure of the protein? You need it so that you can understand its functions, because certain um, proteins which have certain functions have a particular structure associated with them. For example, you have a protein which no one knows the function for, then but you only know the structure for it. You can compare it with existing proteins for which the functions are known to find out what are the commonalities, and then you can figure out that yeah, this protein might be responsible for this particular function. So, proteins function as enzymes. For there is protein ligand binding. They function as metabolic intermediates. They act as cell surface receptors. They function as for phagocytosis, antigen presentation, antimicrobial killing, essentially immune action, inflammation, tissue repair, and remodeling. So, a lot of the proteins are known for catalyzing chemical reactions, which are responsible for running the metabolism, manipulating DNA, replication, transcription, translation. Uh, these There are thousands of reactions running inside a human body at any point, and most of these reactions are catalyzed by enzymes. So uh, some of these enzymes can catalyze the reaction to enhance its rate as high as 10 to the power 17 folds. So, enzymes like OMP carboxylase can run to 10 to the power 17 folds of rate enhancement. Staphylococcal nuclease 10 to the power 14. AMP nucleoside 10 to the power 12. So, all of these, even the lowest carbonic anhydrase can run till 10 to the power 6. If they were uncatalyzed, if these reactions were allowed to run uncatalyzed, the rate of reactions will be very slow. They'll take seconds, months, years. Therefore, the metabolism of human body will be slowed down a lot. Life as we know it won't happen, essentially. Next is reversible binding of a protein to ligand. So, depending on the folding of the protein, they have a defined 3D region wherein the protein can interact with other cellular molecules. For example, in case of oxygen and CO2, which is done by hemoglobin or myoglobin, both of these molecules. So myoglobin is present in muscles, where hemoglobin runs through blood. Both of these molecules have active sites to bind to oxygen. Hemoglobin is present inside the RBC and at the lung surface. It will bind to oxygen and it will release oxygen in a controlled manner, to not in a very disturbed manner, but in a controlled manner, to provide oxygen for running cellular metabolism. At the tissue site, it binds to CO2, it releases it to the atmosphere at the lung surface. So all of this happens by hemoglobin. Inside of muscle tissues, myoglobin is present, which can take up the oxygen from hemoglobin and then use it as needed. So, in a normal gaseous exchange, the outer air has 21% O2, 0.04% CO2, around 75%, 78% of nitrogen. But that does not matter. So, in the lungs, you inhale the air, it goes into the lungs. There is an exchange on the alveolar surface. So, the basic subunit of lungs is alveolus. So these are essentially air sacs on the surface of which gaseous exchange can happen. So there is a lot of capillaries running through these air sacs. At the alveolar surface, venous blood which has less oxygen, more CO2 will come in, come in contact with the alveolus, exchange the gas, and then uh, go back as an arterial blood or oxygenated blood, which will have less CO2 and more oxygen. Then these can go to other places which have low oxygen, release the oxygen there, carry in the carbon dioxide from that place, and go back into circulation. The reversible binding of protein to a ligand also helps it to function as an intermediate for as a metabolic intermediate in so enzyme can can accept different metabolic intermediates to run the metabolism. It will have an active site, which is this in this enzyme's case, wherein the free substrate is going to come and bind. This free substrate, there are a lot of hypotheses on how this happens. Then this enzyme substrate complex undergoes various changes and results in an enzyme product complex. This can be by using some energy, by some pump, but it requires if the difference between the substrate and product is negative, then it will require some energy to make the enzyme product complex. Then the enzyme product complex can dissociate. So the binding between enzyme substrate and enzyme product is very different. Then the enzyme product complex can dissociate, and then you have a free enzyme and you have the free products. This free enzyme can then go back here to function again. Okay. Proteins proteins are present at multiple places within the cell. On the cell surface, they can act as receptors, where they can bind to different ligands present in the inside the cell or outside of the cell and mediate various functions. For example, LDL receptor can bind to oxidized LDL and remove the lipid from circulation. In certain cases, these um, receptors can go back into the cell, get recycled, then come back to the surface after delivering the ligand to the storage system, the endosomes or lysosomal systems. This is also so other receptors can bind to other molecules, then take up those molecules inside in a vesicular or in a non-vesicular manner. A lot of immune action that happens in the body is because of proteins. Immune action can be of different types, but the end result is killing of the microorganisms. So if inside the body there are pathogens or unwanted debris, then it can be scavenged by the whole host body by phagocytosis, wherein cells such as macrophages, which are immune cells, extend their essentially extend their cellular membrane. Pseudopodia is the cell membrane. So if this is a cell, the cell will send out projections like this, known as pseudopodia, which can engulf the foreign particle and entrap it into a vesicular structure known as phagosome. So if this is the microbe or a foreign particle, they will form a covering around the foreign particle. This foreign particle phagosome complex can then fuse with lysosome to form phagolysosomes, which will so lysosome have a lot. If you remember the class where we had learnt about different organelles, lysosome have a lot of hydrolases. They have a very destructive microenvironment, because of which they can they can digest different uh, they can digest different uh, complex sequences to form smaller peptides. So after engulfing the foreign material or the microbe, the formation of phagolysosome leads to destruction of the material into smaller peptides, which can, if needed, be utilized by the body. The Apart from phagocytosis, microorganisms can also be killed by reactive oxygen species such as superoxide, H2O2, hydroxy radicals released after a respiratory burst. These will essentially bind to the these will essentially attack the microbes, because microbes might or might not have the mechanisms in place to combat such reactive oxygen species. Usually they die after exposure to such reactive oxygen species. Okay. uh, these apart from so after the digestion of the foreign material by the phagolysosome complex, the digested the components of the digested microbes can be can be presented outside of the macrophage, so that lymphocytes can recognize the uh, constituents of the microbe. This particular process is known as antigen presentation, wherein the macrophage or other immune molecules present is the possible what is the a fragment of the microbe, so that there is a memory in B or T lymphocytes for uh, especially in T lymphocytes for when there is another attack by the microbe. Okay. Macrophages, which have been exposed to bacterial components or interferon gamma, activate a wide array of inflammatory molecules to inhibit or kill the pathogens. They essentially produce lot of reactive oxygen species to oxidize the pathogenic organism, and the point of inflammation is to reduce microbial burden in host. But in certain cases there might be an excess inflammation, or there will be a bystander effect, wherein some of the host tissue is also getting damaged because of production of these reactive oxygen species, and this might disturb homeostasis. If there is a under or over production of inflammation, both of these will affect the homeostasis of the individual. Macrophages also play an important role in wound healing, tissue repair, and remodeling. If in at the wound site there is injury, there is an accumulation of lot of dead cells, cellular debris, which will be phagocytosed by macrophages. uh, Macrophages will also secrete components such as transforming growth factor beta, which is a protein, platelet derived growth factor, fibroblast growth factor, insulin like growth factors. This will stimulate production of various components which is responsible for sealing the wound. Some of these components are extracellular matrix proteins, which are present between the cells within the body. One such example is collagen. uh, so at the site of the injury, macrophages will stimulate production of collagen from fibroblast cells, uh, and then they so there is a constant remodeling and repair going on at every site. If there is a lot of collagen production, then mmps can also secrete macrophages can also secrete matrix metalloproteinases, which are responsible for degrading the collagen, and this can help fibroblasts and epithelial cells to migrate at the wound site for new blood vessel formation after injury. Okay, so now we know what are the different functions of protein, what are the different function of different proteins found inside the body, but how would you measure the amount of protein in a biological fluid? uh, ELISA is one such immunological technique used to measure the level of antibodies or protein in a body fluid. It is useful to identify the antigen or cross reactivity of an antibody. This can be performed in many ways. Um, there can be direct ELISA, wherein your antibody, which is already tagged, binds to an antigen. When you put the substrate, then uh, there is a colorimetric or a fluorescence signal that you can quantify. There is indirect ELISA, where to the antigen you have one antibody binding and then a secondary antibody, which is a conjugated antibody. So when I say conjugate, it implies that this antibody is bound to something else which can produce a signal. This conjugate can be a fluorophore, which if you put under an appropriate wavelength will lead to production of fluorescence. This can be a luminescence based conjugation, where there are examples such as hydrogen peroxide. So if you have a hydrogen peroxidase, then there will be you can put peroxide and this will give you a signal. So it all depends on what kind of fluorophore or conjugation you are using. There is something known as sandwich ELISA, wherein you first capture the antibody using a already coated plate, then you uh, use an use indirect ELISA to know how much is the antigen concentration. Certain studies use competitive ELISA to find out how much is the binding affinity of a particular for a particular antigen, wherein they have an inhibitor antigen and the actual antigen, and they do a serial dilution of all of these components to know how much is the binding affinity. So first, coming to indirect ELISA, it is used to measure the level of antibodies in serum and calculate the titer of the antibodies. So a known amount of antigen is coated in the well and is incubated with different dilutions of the antibodies. The antigen bound is then recognized by a secondary antibody linked to an enzyme. If you add the proper substrate, then a colorimetric substrate can be used to measure the level of the antibody. If you okay, so there are multiple things that you can change. You can either keep the antigen amount constant and then add different amounts of antibodies, or you can add different amounts of antigen and you can keep the antibody level constant. What is required in all of these situations, that you draw a standard curve. What is a standard curve? It's essentially you add known volumes of, so if this is your concentration of the antigen or the antibody and this is your fluorescence, you know how much fluorescence will come from each of these concentrations. So you get something like a curve like this. Then when you have a antigen of unknown concentration, you can run an ELISA for it and then place where exactly in the curve the concentration lies. Okay. The materials and reagents required for performing an ELISA: you need a bicarbonate buffer of pH 9.2 to pH 9.6, depends on the kit. Filter sterilizer using a 0.2 micrometer filter, so that you have a debris free solution. You need a ELISA plate, which is essentially a flat bottom clear 96 well plate. antigen solution which can be prepared in the bicarbonate buffer or the coating buffer. BSA solution, you need a primary antibody, secondary antibody, and PBS containing twin 20. So the procedure is that if there is a container coated with protein, you allow the antibodies to bind to the protein. You wash off the excess antibodies. You then add your secondary antibody, which will bind to the first one, which will have the conjugation for the substrate. When you finally add the substrate, there is a visual change that confirms whether the protein is present or not. So the procedure is you coat 5 mg per ml antigen solutions in bicarbonate buffer, dispense 50 microliter of each into a microtiter plate and put it overnight for coating. Then you block each well with 1% BSA. So what blocking does it prevents non-specific interactions between the primary antibody and the antigen from happening. It binds, so the interactions between the blocking reagent and the antigen will be weak. If there is a stronger affinity of the antibody to the antigen, then only it will replace the blocking reagent and place itself. Then you prepare the primary antibody dilution and then you dispense each of these dilutions into the respective well, incubate it for a particular amount of time. In this case it's 45 minutes, 37 degrees Celsius. This might vary depending on your antibody, antigen, etc. Wash it 4-5 times with the watch buffer, which is PBS and 1% twin 20. Then you add the appropriate dilution of the secondary antibody, which is constant for every well. Then incubate it again, washes again. Then you dispense the colorimetric solution, the substrate in its substrate into the plate and stop the reaction and take an absorbance reading. So you will get something like this. To perform sandwich ELISA, this kind of setup is used to measure the level of a known protein in serum. So what you do is you add a known amount of antibody specific antibody or the capture antibody to capture the antigen. This antigen is then recognized by the secondary antibody linked to an enzyme, which can be then measured using a colorimetric substrate. So the capture antibody will collect the antigen from the sample. Then a second antibody is used to detect the antigen bound to the capture antibody. Both of these should have different sites. This secondary antibody may or may not be conjugated. If it's not conjugated, you need a third antibody. If it's conjugated, then you can add the substrate and then get a readout or a signal detection. What are the materials required for this? First thing is a capture antibody. If you want to see TNF, the amount of TNF present in blood, then you use a TNF monoclonal antibody. So depending upon where the antibody is raised, it will have a particular reactivity to different other antibodies. So usually antibodies are raised in mouse, in rabbit, in goat. So when we want to know so in this case, wherein in case of indirect ELISA, if we are using a antibody raised in mouse, then we need to use an anti-mouse antibody as a secondary antibody. If we are using something raised in rabbit, then we use a anti-rabbit antibody. Okay. The capture antibody can be diluted at a particular ratio in the coating buffer. Then comes detection antibody, which in this case is a biotinylated anti-mouse TNF monoclonal antibody. This also has a particular dilution depending on the kit. Then you need the enzyme reagent. Because it's biotinylated, you need a streptavidin horseradish peroxidase conjugate. And then you also need to run standards. So essentially you are adding known concentrations of TNF, and then calibrating according to that curve. Then you need a streptavidin HRP conjugate, the enzyme reagent. You add it to the standards, you add it to the antigen, and then you take a reading. Other materials are coating buffer. In this case, it's 0.2 molar sodium phosphate, 6.5 pH, acid diluent, which is PBS with 10% FPS, wash buffer, which is PBS with 0.05% tween 20, TMB and hydrogen peroxide, which is the substrate solution, stop solution 1 molar H3PO4 or 2 normal H2SO4. Either of these can be used for this particular kit. 96 well ELISA flat bottom plates, a microplate reader at 450 nanometer, micropipettes and tubes and plate sealers. So the specimen should be clear, non-hemolyzed and non-lipemic. If they have fat or broken RBCs inside them, this is going to affect the readings later on, because they'll affect the colorimetric, the amount of color produced. In case of cell culture, remove any particulate material by centrifugation, and usually assays are done immediately. If you are storing, store at minus 20 or minus 80 to avoid the degradation of the components, and usually avoid freeze thaw cycles. If the experiment is supposed to be performed in multiple rounds, a good idea is that you make smaller vials, smaller aliquots, and then freeze them and thaw them as required. In case of patient's blood, use a serum separator tube to allow the samples to clot for 30 minutes and then centrifuge. So you removed all the blood cells and you use the serum. Okay. Then you prepare the standard solution, wherein you put the known concentration of TNF alpha and take a reading of the standards. So because they are of different color, different amounts, each of them will give a different color. Third plate step is ELISA plate coating, wherein you dilute the capture antibody, add it to each well, incubate overnight so that the antibody gets adsorbed to the well. Then the step 4 is blocking, wherein you add blocking buffer to prevent non-specific interactions. Then you add your standard or sample into each well. You incubate for a particular time, take out the sample, wash the plate. Then you add your enzyme reagent. You add a secondary antibody, enzyme reagent, essentially the substrate solution, then the stop solution. Then you take a reading at 450 nanometer. So this 450 nanometer is the excitation wavelength. You also, in this case, in case of HRP, you also take a reading at 570 nanometer to subtract the background. So you get something like this. You negate out the background in each case. You draw a calibration curve. Then so essentially you plot a standard TNF alpha concentration against absorbance. Then you know what absorbance you got from the from in your sample. Then you can compare it with this curve to know where exactly what concentration your TNF alpha is. Okay, are there any questions till now? Okay, fine. I will discuss some questions from related to this week. Please stop me if there are any questions or concerns. Okay, the amino acids are classified based on peptide bonds, pK of their functional groups, chemical nature of their R group, number of hydrogen bonds that they form. The answer is the chemical nature of their R groups. Depending on the chemical nature, they can be aliphatic, aromatic, basic, acidic in nature, negatively charged, positively charged in nature. The following methods are used to which of the following methods are used to analyze amino acids? Polymerase chain reaction, thin layer chromatography, northern blotting, southern blotting. The answer is thin layer chromatography. All of these other methods are used for nucleic acid analysis. The significance of pKa values in amino acids is that they are used to calculate the charge of amino acids at a specific pH. They determine the color of amino acids. They dictate the binding sites for peptide bonds. They determine the aromaticity of the amino acids. The answer is they are used to calculate the charge of amino acids at specific pH. So if you were to put different amino acids at different pHs, you find that they adopt different conformations, and this is dependent on the pKa value of the amino acid. The principle behind calculation of RF value in TLC is ratio of analyte distance to the solvent front distance, ratio of solvent front distance to the analyte distance, molecular weight of amino acids, or the pK of the analyte. The answer is ratio of the analyte distance to the solvent front distance. The property of peptide bonds that restrict rotation is the high energy of H rotation, interactions with side chains, partial double bond character, or hydrogen bonding with water molecules. The answer is partial double bond character. This dictates whether they have what dihedral angles they can have. Amino acid isolated from cheese and named accordingly is glycine, tyrosine, asparagine, glutamine. The answer is tyrosine. Tyros means cheese. Secondary structure that is characterized only by intramolecular hydrogen bonds forming between nearby residues in the chain is beta sheet, beta turn, alpha helix, random coil. The answer is alpha helix. So they have the R groups are pointed outwards, and there is a bonding with intramolecular hydrogen bonds forming between nearby residues. The feature that differentiates parallel and anti-parallel beta sheets is the presence of disulfide bonds, number of peptide bonds, the type of amino acids involved, the direction of the polypeptide chains. The answer is the direction of polypeptide chains. In parallel beta sheets, these polypeptide chains run in parallel, and there are hydrogen bonds between these sheets, whereas it's the opposite in antiparallel beta sheets. So in case of beta sheets, strands of beta sheets will have hydrogen bonds in between them. However, in case of alpha helices, nearby residues have hydrogen bonds, because of which the structure is different between these two. The method to identify N-terminal amino acid of a protein is thin layer chromatography, Edmond degeneration, Ramachandran plot, STS page. The answer is Edmond degradation. The other option is Sanger sequencing. Non-experimental method used for modeling protein structures is gel filtration chromatography, NMR spectroscopy, SDS page, homology remodeling. The answer is homology modeling. You can use gel filtration chromatography and STS page to know what is the size of the protein, but not really the structure. NMR spectroscopy can be used for knowing the structure. The chemical treatment used to break disulfide bonds during protein sequencing is cyanogen bromide, dancyl chloride, beta mercaptoethanol, or trypsin. The answer is beta mercaptoethanol. Otherwise, you can use DTT or perfluoric acid. The role the proteins play as enzymes: synthesizing amino acids from DNA, storing genetic information, transporting lipids in blood, catalyzing chemical reactions by lowering activation energy. The answer is catalyzing chemical reactions by lowering activation energy. Dynamic light scattering is useful in studying protein ligand complexes to evaluate the size distribution of the aggregates, measure molecular weight, monitor heat changes during binding, or quantify enzymatic reactions. The answer is to evaluate size distribution of protein ligand aggregates. In protein ligand binding, a low Kd value indicates weak binding affinity, strong binding affinity, irreversible binding, or no interaction between protein and ligand. The answer is strong binding affinity. Kd means dissociation constant. So if there is a low Kd, implies there is a very strong affinity between the protein and the ligand. If there is a high dissociation constant, then the ligand protein complex is prone to dissociation. The parameter that is directly measured by isothermal titration calorimetry is heat changes during protein ligand binding, fluorescence of the ligand, molecular weight of protein, or the velocity of enzymatic reactions. The answer is heat changes during protein ligand binding. In case of ITC, what you do is you put a reaction inside a calorimetric chamber, then you measure how much is the heat produced from that heat produced or taken off in that reaction, and therefore you can measure the energy of the reactions. Are there any questions? Please let me know if there are any questions. Okay. Then this is, if there are no more questions, then we can end the session here. Thank you for attending.